Thanks so much again. I listened to Jonathan talk, and just to start the discussion, one of the topics that is, I think, nice to break the ice is the placenta microbiome. Jonathan mentioned it a bit during the talk, but maybe it's the time also to we can take a few minutes to expand a little bit on that. So I don't know if maybe Jonathan wants to start also here.
Well, I mean, I could. You probably know more about it than I do. I mean, I think the main issue, which I can just talk about, the general issue is that whenever you're working with indirect studies of some system, like you doing PCR and DNA sequencing to study communities of microbes, you run into an issue, which is that the method can create artifacts. And you can have artifacts created in all sorts of different places. Some are more appreciated now than they used to be, like the issue with molecular biology reagents coming with DNA, right? I mean, so when you grow up an organism to make an enzyme that you then sell in a kit, or when you make water to put into the kit, or you make some buffer to put into the kit, that can come with DNA. And when you push the frontiers of trying to detect microbes that are in very, very low abundance with some amplification method, you run a big risk of amplifying things that are not from your sample. And there are also many other steps where laboratory, challenges can come into play. So you have to be really careful, especially again when you're working with systems with low abundance of microbes. And people have known about this for forensics, for ancient DNA studies, for all sorts of other studies, and it just unfortunately has reared its head in some microbiome studies. And it seems reasonably likely that many of the claims of a robust placental microbiome are more due to contamination issues at different stages of the process than to the existence of living, viable microbes in functioning placenta. I mean, it doesn't mean that sometimes placentas, people can get an infection and you can get microbes into a particular place where they're not supposed to be. But a healthy placenta, most of the studies I've seen suggest that there really are no viable microbes. You're on mute, Rick.
Thank you. I totally agree with everything Jonathan said. We've published a couple of papers in this area. Right after the proposal for the placenta microbiome came out, some of my colleagues here at Penn with running a preterm birth institute couldn't ignore the proposals. So we did a very careful sampling study, and we couldn't see a placenta microbiome over background. We could see a little bit of microbial DNA, but not over background. And we did the experiment twice with two different kits. And in each case, the placenta microbiome resembled the background, but it was different background with the two different kits, which was one of the important indications. And there's since been very large studies from the Sanger Center and others getting pretty much that result. And the placenta microbiome is an interesting case because you have a separate line of experiments which suggests that the womb is normally sterile from germ-free animals. You can do a sterile cesarean section, get the pups, move them into a sterile isolator, and they stay sterile, which would seem to be, a definitive functional experiment. And that's even been done with humans. So to me, that in rare cases, like in the 60s, they delivered babies sterilely and they stayed sterile. So I completely agree with Jonathan that this is instructive case. And, as reviewers in papers, I'd urge everybody to ask people to please publish their negative controls, be really clear about what they did exactly and how they, in a low biomass setting, how they distinguished what they're reporting on from the reagent background.
Yeah, if I can comment, it hits 2 points because about the communication, it is so harsh, this placenta thing. It's not going away. After it was published, it keeps on singing around. People are still referring to the placenta microbiome. So that's also the overselling. And then if it's out there, it's hard to get rid of it, even if the science is good. And the other thing that reminded me of was at the beginning of sequencing era that we had all these papers about the rare biosphere, where it was actually just sequence errors, but it seemed like the microbial world was like ever expanding. And yeah, this is also what we have to deal with, right? Yeah, there's
Yeah, no, I think it's great. Overselling the placenta microbiome. Sorry, Jonathan, I think you're muted.
No, go ahead.
Okay. No, I was saying that overselling the placenta microbiome is something easy, no? Because it will be something nice to hear. So probably overselling correlates a little bit with what we would like it to be true, no?
I've given out many overselling the microbiome awards to placental claims.
and one of the problems was also that they made these correlations, and then with random correlations, it correlated to the oral microbiota, which then, it just all went sort of in the wrong direction, because you need to find something, and whatever you provided, something will be the closest hit, right? Yeah,
that was one of my earlier placental microbiome overselling awards, was about the oral microbiome putative connection and how this explained everything in human health.
So, and then what is your guys' opinion then on so there's a breast milk microbiome, but there's two opinions there. One is that it's actually transmitted from gut to milk, and the other one's that it's coming from the outside. I don't see the good evidence there either yet.
Yeah, we helped Grace Aldrovandi with a study there, and we did find that samples of women's breast milk looked more, there was more resemblance to their baby's fecal samples than to some other randomly selected baby fecal samples. So one possibility is that there's organisms in breast milk that are transmitted directly to baby, but it could be the other way, or it could be coming from some third site. It's not, I wouldn't say it was definitive by any means, but there's some suggestion of some amount of sharing, although, exactly as Jonathan's been saying, what that correlation means is could have a lot of different origins.
Too bad we don't have Maria Carmen on the panel,
Yes.
because she has a lot of experience into that, of very nice science, actually. Yeah. And, both Rick and Clara Sensors called also the negative results, no? That we need to report negative results, because especially in high-dimensional field like ours, if you report only the positive results, maybe overselling them a little bit, then you create a huge bias in the field.
Yes.
What can we do here?
No, I mean, I think that's been a struggle in many areas, right? In many fields, stem cells, cancer, any area, the many people don't publish the negative results that they get. And it's hard to publish them. There have been journals created that are explicitly focused on negative results, but you just don't get a lot of credit in the current academic world for doing that, and we need to keep working on it. I don't think it's been solved. I don't think that people publish this enough, and I don't think that we give them enough credit for it. So I think it's definitely an area that scientific publishing needs to keep working at.
But there are the preprint servers and stuff like that. You can put up data on preprints. There was just a paper that came out. There were these wild claims about SARS-CoV-2 in deer and like animals that showed huge numbers, huge percents of infection. There was just a paper that came out of a Canadian study of like 1,000 samples where they didn't find a single example of SARS-CoV-2 in any animal that they studied. And so that seems like a pretty useful negative result. And that came out on a preprint server. So
yeah, that's a really good point. I mean, I think that the preprint servers do serve that role as long as we get more people to actually post them there.
Yes, and I think if I want to be a little bit more provocative here, do you think that this problem of reporting negative results is particularly big as a problem in the microbiome field compared to the other fields? This is coming also from a question from the audience right now, that is asking whether the hyper recognition and the overhyping of stuff are particularly bad in the microbiome compared to other genomic fields. What's your
No, I mean, I love this question. I mean, first of all, I should say that I used to give out an overselling genomics award on my blog and in talks, and then that evolved into the overselling the microbiome award. Genomics, in many areas of science, obviously have this problem. Genomics had a big problem in this area and still does with, originally the Human Genome Project spending in the US $2 or $3 billion of federal government money to sequence the human genome. And literally there were people in Congress who claimed that this was going to cure cancer, that if we just spent this money on this, it was going to cure lots of human diseases. And I'm not kidding. There are, after the human genome was sequenced, after the big press conference where they announced the public and the private human genome effort, there were hearings in Congress where they called people up and said, okay, you told us you were going to cure cancer and you haven't. WTF, we don't want to give you any more money for this type of thing because this has been a problem. And so it's not just the microbiome area, but the microbiome area is ripe for this in some sense compared to other areas because it is so complicated. Communities of microbes and how they interact with a host are so complicated that it's very easy to get statistically spurious results without even having a real correlation. You can think you have a correlation with a big number system. And, if we say this all the time, like if you're looking at 10,000 taxa and their relative abundance across different samples and you have 10 patients, 5 with the disease and 5 without, you literally are guaranteed to have perfect correlations to that trait. Statistically, you will find them. That doesn't mean they're real. And then with the correlation and causation issue, you now have on top of that real correlations, but that have nothing to do with the cause that you think they're due to. And if you add on to top of that, the complexity of, we're many cases, sequencing fecal samples to try and understand what's going on in a complicated gastrointestinal system. We're running bioinformatics workflows that are not, perfect. I mean, like all of those things added together, it's really easy to find spurious, fake results, and people can either mistakenly present those or purposefully take advantage of it for hype.
Yeah.
This is like, I love, sorry, Clara, how our audience is picking up with our comments, because we just had a comment from Kate Kennedy say that it seems particularly common in microbiome research to choose the method for analysis based on what shows significant results, which is probably also due to the fact that our field is not close to that standardization yet. No, so one can, for the good and the bad, choose the methods based on the type of analysis that they are doing.
I mean, go ahead, Rick.
It takes a lot of strength of character to get a negative result in a microbiome experiment. There's so many different ways to slice the data, to really be tough on yourself, really correct for multiple comparisons in all the way you should. And in order to get nothing, I mean, that's really shows you did a very good job on the analysis if you succeed in doing that, sort of following on what Jonathan said.
Yes, and another thing is, especially if you set up a very clean clinical study, you're also always looking at the primary outcomes. And if the primary outcome is not it, a lot of people start focusing on them, even though there's other outcomes, other metadata that can explain the things that you see in the microbiome. So it was not this certain disease, but it was the diet or the medication, but they forget to look at it because they are only focusing on their primary objective, which means they want the microbiome to be associated with it, but probably the variation is caused by something else. And either they didn't measure it or they can't find it, but people stop looking.
Yes.
I think it's really important to be tough on yourself, correct for multiple comparisons, and say if there's nothing that survives at the end. That said, though, I think you're within your rights then to say, we get nominal p-values that are significant in this, and this respect, because those can be hypotheses for the next study. Just be careful to say that after you correct for multiple comparisons, there's nothing there, and it may not mean anything.
Yeah, and I totally agree, both with the positive and negative points that Rick just raised. I mean, I think that as a sort of natural historian, I think it's important to not always rely upon hypothesis testing for every single thing that you do. But going back to the point from Kate, is that you should think about your methods that you're going to use before you actually do the analysis, rather than constantly changing them during the course of the analysis. I mean, people do this in clinical trials, right? You register in advance the methods that you're going to use to analyze the data. People don't do that very much in most other fields, but it's not a crazy idea.
Yeah,
it's quite solid. Yeah.
And I have, however, a very good news for all of us, because a comment from Roberta Raffaetta. She says she's a social scientist, and they have the same problems with negative results there. So at least this is slightly reassuring.
Yeah. It's a huge issue in clinical science, drug testing, stuff like that. You have a publication bias that can be quite strong. There are even techniques for trying to figure it out. So if as your sample size goes up in different studies, you get closer to the null hypothesis. That's telling you maybe there's another side to it that was never published and you don't have in the analysis.
Yeah. And another point maybe that we raised in our private discussion is that as soon as you start the statistical analysis, you started with a microbiome, but then people start all of a sudden focusing on ASVs and OTUs and single microbes that should explain then your results, even though you were looking at a bigger system. And I understand that's the easiest to find a correlation with a single microbe or an ASV, but it's probably not it. It cannot have so much impact on the complete system in the microbial ecological sense.
Definitely. So I think here the questions are probably two in this on this point. So the first one is, how can we model the overall ecology and behavior of the community in these studies? But the second is also, given the complexity that SVs, OTUs, but also single microbes and single strains are adding to the picture. A question from Ami Manges is pointing out, how can we design studies when we have to correlate such complexity with respect to a lot of complex readouts and correlating and confounding factors. So two big questions, probably cannot answer comprehensively to that, but what's your comments on that?
No, I mean, I think it's a great question. I don't think people have an obvious solution to it. I mean, I think that another thing we haven't really raised here that is important is to have a full and, as possible, a reproducible description of all the methods that you use, so that other people can run them on their data sets or rerun them on your data sets in some way, and trying to get around some of the issues of like, if you apply some set of methods to one data set, it would be really interesting to apply them to another data set that maybe you know the answer to already, or maybe you know a little bit more about the biology, like you were mentioning with the placenta negative sterility issue. So I think that doesn't sort of answer the question directly, but the more that we put out GitHub packages to reproduce bioinformatics workflows. And I'm reminded of Pat Schloss has had a few papers where he publishes the entire code, starting from downloading the data from GenBank or somewhere, where that you can reproduce the entire, literally producing the figures for the paper from the entire set of code. And you could rerun it if the data changes. You can rerun that entire thing. So I think that that's a component of this. As for, it is very complicated. There are a million methods to use, and it's not obvious which ones are the best ones to use. So I don't know of a great solution.
I totally agree, but don't be paralyzed. Form aggressive hypotheses and go for it. Do exciting research. One thing you can do once you have an interesting result is a validation cohort. Just do it all again separately and see if you get the same answer.
Yep.
That cuts through a lot of different kinds of concerns about the methodologies.
Yeah, it's not only, I can give you the example, we have a couple of papers in which we co-published these two papers with other groups, which basically did the same thing in parallel to us. We didn't know it until close to the end. And on just only partially overlapping data sets with different methods, we achieved the same results. So we were very happy that the results were basically the same, and also that journals recognized the power of showing two papers side by side, different authors, different methods, and partially different data saying the same thing. I think that I was very happy about that. Clara, you wanted to add something? Sorry, I interrupted you, I guess.
No, I think to add to there is still that we should also focus on real microbiology, microbiophysiology, and grow the bugs that we can grow the communities if we can grow them, and see what they're actually doing, even though it's hard and it's outside the human body or outside any animal body, but it will help us to predict and to do interventions and to see what's actually going on.
Yeah, agree. And on the second part of the question, though, we don't want to scare people, no? So it's still possible to do great research and great, even clinical trials. But what can we answer to the question on how many things should we measure around the microbiome, which are the most important? So guidelines around the study design, I guess the question is. Because then, if one is not expert, is not so nice to tell them at the end of the day, well, you should have done that. How can we help promoting good study designs?
I mean, I think this may have come up in the chat a little bit, but there have been these sort of standards working groups that have been thinking about, it's not really this, it is partly the study design, but it's also partly thinking about how you record and report associated, quote, metadata with various studies. That includes, the design, the sampling, the informatics, all the other things associated with a particular study. I mean, that doesn't solve the issue of study design per se. So I think that you need to really, I mean, this also was raised in the chat by someone, DNA sequencing, which has been used by, it's not, of course, the only way that we study communities of microbes, but it's used by a lot of people. It's become so cheap and easy that anybody can do it in some sense. And that's good and bad. The good is it's expanding the areas that people can look at things. The bad is that the study design, it's frequently just, you're doing some study of something else and you throw in the microbiome at the last minute to, grind up a sample, extract the DNA and do some sequencing. And you may not have collected the right information. You may not have designed the study in a way that prevents contamination or that, so like, I think that it's great that it's expanded access to the field to lots of other people or to some areas, but it also comes with challenges. And this goes back to something I think Clara may have been mentioning, which is, do real microbiology too, right? I mean, like they actually studying the microbes, the functions, the metabolites, the things that are being produced in the system is really, really important. And so again, it's not that everybody just does DNA sequencing to study microbiomes, but it is the low-hanging fruit for a lot of studies. And following up, some observation that you make, like Rick was saying, a hypothesis that you generate from this, to actually testing it with experimental studies is really important.
Yes, and if I can comment to that, so a lot of time people come to me with data or with samples, and they just treat the microbiome as a black box. So let's put something in and we'll get something out without a rationale of what microbes might actually contribute to whatever they want to look at. It's just that there's a microbiome, it will do something. And I think there's a nice example, and it was a negative result, and it was published really high impact in gut, where they did antibiotic in the bloodstream, and they thought, well, antibiotics, they impact microbes, right? But it actually didn't, because the antibiotics did not flow enough into the gut probably to affect the microbiome. And it was really, this was a nice example of a negative result that completely made sense at the end.
Yeah, I think what you need in experimental design, sort of what's allowable, is determined to a great degree by what your goals are. Much as you folks have been saying, if it's fine to do exploratory stuff, natural history, trying to sort of set up the ideas for the next experiment. And then but the other extreme would be, a clinical trial where you're asking whether an intervention does something to, improve people's health. And there you need to write down, as we've been saying, your statistical plan, your sample size, power analysis, blah, blah, blah. Really, really rigorous setup. But I think there can be another extreme where you're just trying to learn something new about a deep sea vent organism that's never been studied or whatever it might be, and just collect natural history data to advance your thinking in some unformed kind of way.
Yes, absolutely. And also in the chat, they were mentioning the STORMS checklist, which is a large recent effort by many researchers led by Levi Waldron to try to strengthen and organize the reporting of microbiome studies. That can be helpful, and I think several journals are already adopting it, so it's a good sign. But let me change slightly topic here. So I like that Clara mentioned that when we study microbiomes, we should have a microbiological understanding of what we are looking at. But what about the immunological viewpoint? Of course, just for the human microbiome, but sometimes probably the microbiome should be seen as the other side of the coin with respect to the immune system. But it's not particularly easy, and maybe it's our fault to interact and design studies with immunologists. What do you think?
It's sure that a lot of the importance in cases where the microbiome is doing something involved with human health will have the immune system as a part of the story. And there probably are things where just beginning to dawn on us that would be interesting to study, like immune responses to bacteriophages and all kinds of stuff like that. So I think it's a huge area and something that's sort of conspicuous in its absence in many kinds of microbiome studies if you don't do a good job of it.
I mean, I think that in general, what I sort of hinted at in my talk, which was that host microbe interaction studies where there's a single microbe involved, like a single pathogen or a single well-documented mutualistic symbiont, people have gotten to a point where those are really detailed studies of the interaction between the single microbe and the single host, and you can study the developmental processes and the immunological processes and so on. And those have been really exciting. With communities of microbes, it's a lot more complicated, and therefore I think that part of the issue has been that it's so complicated that people focus in obviously on one part of it, which is like just the bacterial portion of the microbiome in fecal samples, or just the fungi that are on the skin of some individuals or something like that. And it's too complicated almost in some sense to close the loop and have a full interaction between the host immune system or the host gene regulatory system or whatever, and the community of microbes. But it's clear that we have to do that. So I think that, building up to the point, I mean, obviously there are some great studies that have been done, in particular in model organisms, of starting to do this. But we really need to expand our ability to study systems as opposed to one portion of the system.
Yeah, which is a question that also can go into intervention, no? Because focusing on single bacteria means traditional probiotics, if you like, whether they work or not. That is the idea, against the fecal microbiota transplantation, that again may work or not work, but is the system level idea of modulating the microbiome.
And the combinatorics becomes really hard, right? I mean, like, if you want to study 10 microbes and vary their relative abundance and vary their presence and absence across the system, you're done for the next 400 years for experiments. So I don't know the ideal solution to this because it becomes so complicated, but clearly we have to continue to work on communities. And I was mentioning this, I think, to Clara in the chat earlier, like, in tropical rainforest studies and tropical ecology studies, nobody would say that if all I have to do is preserve, one parrot species and then introduce it into new places and I'm going to protect the rainforest. And that is somewhat what people are doing with probiotics, but it's also very, very hard to study a tropical rainforest with 200,000 species in it. And
it's not only the animal species, in that case, and I think in the fecal microbiota transplantation case, we are transplanting also, metabolites, antibodies, a lot of things that may have actually an impact as big as the microbes themselves.
Yeah.
Well, it's not totally hopeless. You can hope that an experimental program might lead to proposals for specific metabolites that do stuff. Then you can test those metabolites separately and see if they have a function. And of course, there have been many high-impact papers that have run down a microbiome effect to something more specific and then studied that, which is pretty cool.
Yeah, agreed.
Absolutely.
Yeah. And
we're running
proteins.
Yes, indeed. We are running a bit out of time, but maybe there is a time for a last question, still inspired by Jonathan's talk, which is the one about, so Jonathan talked about companies, selling, sometimes overselling the microbiome. On the other, I was thinking that if you think about how many samples Ubiome or other companies were able to collect over a short amount of time from such large population, and thinking about the largest microbiome studies that are funded by public granting agencies, we are order of magnitudes away. So companies may have the power to collect so much information, microbiome information, that for scientists is something incredible. So how do you see this? On one side, we cannot, sell something or support selling something that is not scientific. On the other hand, that opens, possibilities, those possibilities that may not be available otherwise.
Yeah, I think it would be, one thing that we've talked about, and there have been efforts for this in cancer studies and other clinical studies, is getting companies to share more of their data when they're not using it anymore, or when they're not, some parts of their data being more open to release of that information, barring privacy issues and other things. And that there's certainly been a big effort for that in clinical cancer studies and other drug studies. I have not seen as much of that in the microbiome space, and that would be a really useful thing. I do think that large-scale studies across massive populations where you try to do things in some controlled manner with the same methods can be really useful. That was sort of the point of some of the citizen science projects that I briefly mentioned, but as Nicola pointed out, the scale at which even those happen is not the same. The US government spent $3 billion to sequence the human genome, and it didn't give us shit. So, hey, maybe they should sequence shit and give us more.
Whoa. Yeah, it's complicated by the fact that the probiotics industry is really pretty disreputable. They're selling products that don't do anything. There's a body of law that protects them that's pretty much outrageous. The New York Times called it the Snake Oil Protection Act. I was an expert witness once when the FTC went after Philips Colonic Health, and they had absolutely no data to say that their products did any of the things they said in their TV ads. But still, they won the case because it was protected by this act of Congress. And so it'll take another act of Congress to change that. And I think that would be a good thing for folks like us to lobby for.
Yes, and on the other hand, if we do find something spectacular, they will pick it up. And then we can influence their huge influence.
Yes, great. So we are really running out of time. I enjoyed so much the discussion, and we will be very happy also to hear from the attendants whether they want this experiment to be repeated in the future with other panels. It would be very fantastic if we have feedbacks by email, by on Twitter, wherever. Let me thank you so much again, Jonathan, Rick, and Clara. That was really great to have you here and discuss. We will have continued for hours. But thank you so much again.